So we've examined the problem of perception at the behavioral level, right? And we noted that if we just look at perceptual behavior, it seems like perceptual experience is a combination of bottom-up information, sensory signals, and top-down information, prior knowledge. We've tackled perception at the algorithmic level, so the second level of Mars analysis. In the last lecture, we looked at Bayesian perception. So the idea here is that we can model perceptual processes using Bayes' rule. And under this framework, perception is probabilistic. It's a probabilistic inference based on sensory signals and prior knowledge. And so we combine these two things. And the weight that we put on prior knowledge depends on how clear the sensory signals are that we're receiving. So if we want to tackle this problem at the final level of analysis, we have to look at how perceptual systems are implemented in the brain. And we're going to do that today in the context of visual perception. So to start, let's just quickly review how information gets from the retinas in each eye to the occipital lobe to primary visual cortex. So we have two eyes facing forward. There's some overlap in our visual field. So each eye receives information from both the left and the right visual field. And what happens is that photoreceptors in the retina transduce light and turn it into action potentials. Those action potentials are sent down the optic nerve. At the optic chiasm, they cross over such that visual information from your right visual field, so everything to the right of your nose, ends up in your left hemisphere. Everything to the left of your nose ends up in your right hemisphere. And so these visual signals are first processed in the cortex by neurons in primary visual cortex. And as we've reviewed in a prior lecture, those neurons seem to respond to basic features of images that we're looking at. So in Hubel and Weisel's famous experiment, they had cats look at bars of light at different angles, and they recorded from neurons in primary visual cortex. And they found that there were neurons that responded specifically to very specific features of objects, such as the orientation of angled bars of light. And that's what we see in this image. This particular neuron that they're recording from really likes light that is oriented vertically. So it fires the most for vertically oriented light. There'll be neurons that fire for light that's horizontally oriented, and neurons that fire for light that's angled. And these feature detection cells have small receptive fields. What does that mean? Well, it simply means that they monitor a very small patch of the retina. So this particular cell that likes bars of light that are vertically oriented would respond to vertical bars of light shone onto a very, very tiny patch of the retina. And so when we say receptive fields, that's basically what we mean in regards to vision. What area of the retina does the cell respond to? So what happens when we move out from primary visual cortex? So if we move out into the cortical areas that surround primary visual cortex and out into the temporal lobe, what happens? Well, in the 1970s, Gross and colleagues recorded from single neurons in the temporal lobe of monkeys. And what they found was that neurons in the temporal lobe didn't respond to basic features of objects. They actually responded to complex objects. So these neurons weren't responding to shapes, but they were responding to complex objects like, for instance, a human hand. So it seems like as we move farther away from primary visual cortex, neurons that respond to visual information tend to respond to more complex, more complete visual signals. And so the idea here is that the brain sort of builds images up from their basic features. So the retina kind of breaks things down into its most, basic components. You can take any image, right, and break it down into angled bars of light. And that's what neurons in V1 like. But as we move away from V1, so as we move away from primary visual cortex into surrounding cortical areas that respond to visual stimuli, V2 is one area, V4 is another area, area IT is a fourth area, we see that neurons in these areas respond to more complex stimuli. So neurons in V2 respond to shapes, neurons in V4 respond to objects, neurons in area IT respond to complex objects, so for instance faces. And what's also interesting is that the receptive field size grows as we move out from primary visual cortex. So neurons in area IT are actually responding to a very large portion of retinal stimulation. They're monitoring a very big part of the retina. And so a face could fall sort of anywhere on your retina, and these neurons will fire for that face. Whereas neurons in primary visual cortex are only monitoring a very small part of your retina. So again, it seems like vision works by starting with basic features and sort of building those features back up into complex shapes. Now, the details of how that actually occurs, we're not 100% sure how that works. It can get quite complex. But what you're seeing here is the start of what's called the ventral stream. And this is a processing stream, a neural network in the brain for recognizing objects visually. You may remember the ventral versus dorsal stream from intro psych. Let's talk a little bit about these two streams now and how they've shaped our understanding of visual perception in the brain. So we now think that there are two streams in the brain, two processing streams, two neural networks. One that's concerned with identifying objects, and another that's concerned with interacting with objects and identifying where objects are in space. A what stream versus a where stream. And this idea that there are two visual processing streams in the brain dates back to this 1982 study by Ungelader and Mishkin. And they were working with an animal model, so monkeys in this case. And they simply looked at how the behavior of the animals changed when they lesioned different parts of their brain. So in one case, there was a temporal lobe lesion, and in the other case, there was a parietal lobe lesion. And they had the animals do two different tasks, an object discrimination task and a landmark discrimination task. And in the object discrimination task, the animal is shown an object like a triangle, and then it's shown two objects like a triangle and a square, and it simply has to recognize the object that it just saw. So I just saw a triangle, now I'm seeing a triangle and a square. If I pick triangle, I get a reward. So it simply has to discriminate between two objects. In the landmark discrimination task, the animal has to learn that there's food hidden near a cylinder. So in this image, we're actually seeing two what are called food wells. So under these sort of purplish rectangles, there would be food hidden. And what the animal has to learn is that the food in this case is hidden under the well that's closest to the cylinder here. So this is the landmark discrimination task. And again, in these animals, they're lesioning, damaging either the temporal lobe or the parietal lobe. So what do they find? So what this bar plot shows is trials to criterion. What does that mean? It means how many trials did it take for the animal to get really, really good at the task? And so the more trials, basically the worse the animal's performance. PP refers to a lesion in the parietal lobe of the brain. IT refers to a lesion in the temporal lobe of the brain. So what can we see for the landmark discrimination task? Animals with parietal lobe lesions, very, very bad at this task. So it took them a long time to figure out that the food is hidden near the cylinder. Animals with the temporal lobe lesions, on the other hand, were pretty good at this task. Animals with lesions in the lateral and medial striate cortex, so remember, medial means near the middle, lateral means near the outside, striate simply means visual cortex. Animals with lesions in these areas were not as good as animals with lesions in the temporal lobe, but not as bad as animals with lesions in the parietal lobe. So it seems as if parietal lobe lesions really messed up the animal's ability to discriminate landmarks, to see where things are in space. Let's look at the object discrimination task. In this case, we basically see the opposite pattern of results, right? Animals with lesions in the temporal lobe, terrible at distinguishing objects, terrible at telling the difference between a triangle and a square. Animals with lesions in the parietal lobe, they seem to be able to do that without any difficulty. So here we're seeing, something of a double dissociation. Damage to the parietal lobe seems to mess up landmark discrimination, but it spares object discrimination. Damage to the temporal lobe seems to mess up object discrimination, but it spares landmark discrimination. So this is the first evidence that there are two processing streams in the brain, a where pathway and a what pathway, a dorsal stream and a ventral stream. So this dorsal stream seems important for determining an object's location, where things are in space. The ventral stream seems important for identifying what objects are. And again, when I say stream or pathway, right, we're assuming that information is processed in stages. So you can just think of a neural network that's responsible for, in one case, determining where something is in space, and in the other case, determining, what is it that I'm looking at. Now, the study was done in an animal model. Do we see the same thing in humans? So we can look at patients who have had brain damage. And Mel Goodall, who's a professor at the University of Western Ontario, studied a patient named DF who had temporal lobe damage. And he famously had DF do what's known as the mail slot task. And so in this task, what you have to do is you're shown a mail slot at different angles, and you simply have to say or match what angle it's at. So you're shown a mail slot at 90 degrees, for instance, and you simply have to draw a line at 90 degrees, or say the mail slot is at 90 degrees. You're shown a mail slot at 180 degrees, you simply have to draw a line at 180 degrees. So very simple task. Obviously, we could all do this. The second aspect of the task involves posting a piece of mail through the slot. And so what's remarkable here is that Goodall found that this patient with temporal lobe damage could not say what angle the slot was at. So basically, and you can see that in this figure here, the patient was more or less guessing randomly. So the patient couldn't match the orientation of the slot, but if you gave the patient a piece of mail and asked them to put it through the slot, they could do it every single time. Very weird, sort of remarkable finding. And if you read the paper, a very cool paper, Goodall points out how weird this is, right? The patient demonstrates strikingly accurate guidance of hand and finger movements directed at the very object she fails to perceive. So there seems to be a dissociation here, a single dissociation between identifying objects and interacting with objects. So you seem to be able to interact with objects even if you can't identify them. Very interesting. And it seems as if object identification, depends on the integrity of the temporal lobe. I have a video clip of somebody who has temporal lobe damage, and I think this really gets at the point. So in the video, you're going to see this man. He's shown some images, and he simply has to say, what he's seeing. And it's kind of a remarkable video. I think it's probably the most interesting video in this class. I apologize for the quality. I saw it in somebody else's talk, and I sort of borrowed it from their talk, so the quality is quite poor. But just watch, and then I'll dissect the video after.
Let me give you a hint.
Okay.
I'm going to give you a choice, and you tell me what this is.
Okay, I can tell you then.
Okay. Is this a clock, a lock, or a telephone?
A lock.
Is it a lock?
A clock, a lock, or a telephone? Okay, let's rule out a telephone. What? A clock or a lock? Clock. Gotta be a lock. Must be a lock then.
Are you sure?
No, I'm not. But I don't recognize it as a telephone.
Mm-hmm. Do you recognize it as a lock?
No, honestly, I don't. But it's just it really looks very indescript to me.
Uh-huh. But your hands know what they want to do.
I know,
with
and
it.
they want to do something.
That's right.
I can see them.
Uh-huh.
I can just picture them, and I know that I have. It's like I'm undoing a combination lock.
Good.
Huh.
Okay, good. We had to work on that one, didn't we?
No shit.
That's okay.
So I'm just going to jump in and dissect that for you. So what were we seeing there, right? He's being shown a combination lock. He's being asked, what is this? He has temporal lobe damage. He can't identify it. He's been given, he's given some choices here, right? It's a multiple choice question: clock, lock, or telephone. Very different objects. Clearly a lock. He can't identify it, but he knows how to interact with it. And if you go back and watch the video again, you'll see that he's actually, with his hands, he's actually making the movements as if he's unlocking a combination lock. So, striking evidence that the brain distinguishes between vision for recognition, vision for identifying things, and vision for action, vision for interacting with things. So in this patient here, in patient DF, we have the case where, the individuals have damage to their temporal lobes, and they can't identify objects, but they can interact with them. What if we see patients with damage in their parietal lobes? Do we see the opposite pattern? And indeed we do. So these patients were studied by Rizzo, Nawat, and Zeil in the 90s. And in particular, they looked at a patient named LM. LM had damage in the parietal lobe, and they studied LM's perception of visual emotion. And they did that using what's called random dot motion. So this is a random dot kinetogram. And what you can do in the lab is you can make the dots move in random directions or make them move in one direction. And typically what happens in normal healthy participants is that if you, make 10 or 15% of the dots move in one direction, we observe motion in that direction. And that's what we see in this bar plot here. So for healthy controls, when about 15% of the dots move in one direction, the controls observe motion in that direction. This patient with parietal lobe damage couldn't do this task at all. So the experimenters had to have, 50% of the dots moving in one direction for the patient to see visual motion. And it didn't matter whether the dots were moving very slowly or very quickly. Parietal lobe damage is associated with impairments in visual motion. And so if we take this patient LM, and we put their data together with the data of patient DF, we see a double dissociation. So we're doubly dissociating object perception and the perception of visual motion. If you have a lesion in your parietal lobe, you're fine at recognizing objects, but your ability to perceive objects in motion and interact with things is remarkably impaired. On the other hand, if you have a lesion in the temporal lobe, so damage to your temporal lobe, you can't recognize things, but you can see visual motion, you can interact with things. So, classic double dissociation. This argues for functional specialization, right? This argues for the existence of these two processing streams that are distinct. Very strong neuropsychological evidence that in the brain, there's a dorsal stream, which is what we would now call a vision for action pathway that runs from primary visual cortex up into the parietal lobe. And there's also a ventral stream, or a vision for perception pathway that runs from primary visual cortex into the temporal lobes. And so this is how we think visual perception is implemented in the brain. And this framework suggests that, we really care about recognizing what things are and interacting with things in our world. And we care about these two things enough that they're actually dissociable in the brain. There seem to be different neural circuits for each of these two processes. Now in the ventral stream, there's a very interesting area that specifically responds to the perception of faces. And this area is known as the fusiform face area, and it's been extensively studied by Nancy Canwisher and colleagues. And in the original paper, which came out in the late 90s, they showed using fMRI that if you look at this brain area, and you can see it in the temporal lobe here, in green, in this MRI scan. If you zero in, hone in on that brain area, and you have participants look at faces versus random objects, you see that when they're looking at faces, the fMRI signal increases, and when they're looking at objects, the fMRI signal decreases. So this brain area seems to be associated with face perception. Now, this is functional neuroimaging. It's necessarily correlational, but we do know that damage in this brain area or near this brain area leads to something known as prosopagnosia. And prosopagnosia is an inability to recognize faces. And people with prosopagnosia can't even recognize the face of a loved one. They have to use other cues, like the voice of their parents or their significant other. They can't recognize somebody by looking at their face. Now, the fusiform face area is kind of controversial because it suggests that the brain has highly specialized modules or processing units that sort of do one thing. suggestion here is that we have one brain area responsible for face perception. Does that make sense? Well, face perception is something that's kind of special. We're very good at telling upright faces apart. Now, you may remember this illusion from intropsych. This is known as the Thatcher effect. And the idea here is that changes in the features of faces are actually very hard to identify when faces are inverted. So you might not notice anything too odd about these two faces, but if I flip them so that they're the right side up, suddenly it's quite clear that the face on the left is horribly wrong. This happens to be Theresa May, the former Prime Minister of the United Kingdom. And this is called the Thatcher effect because the original stimuli was based on manipulations of features in Margaret Thatcher's face. So it seems as if we have some expertise for identifying differences in upright faces. Faces seem unique, despite the fact that they are often actually remarkably similar. Now, are we born with this expertise, or is it acquired? Well, I can ask you this. What is this? What are you looking at right now? Now, some of you may have said dog, and that would suggest that you basically have no expertise in identifying dogs. If some of you identify the breed of the dog, that would suggest that actually you have some expertise in identifying dogs, and you can tell different dog breeds apart. So you've acquired some visual recognition expertise. And the idea here is that the level of individuation that we afford an object depends on our expertise. It depends on our experience. Right? So you might look at the two dogs on the left here and think, those are dogs. Clearly, you can tell the difference between a dog and a goat, and a dog and a banana, but you might not be able to tell the difference between two dogs because you have no specific dog expertise. And so one counter to the idea that the fusiform face area is an area that is specialized for face perception is that it's actually just an area that's specialized for perceiving things that we've become experts at perceiving. And indeed, this area has been studied in bird watchers. So these are people who are experts at identifying different kinds of birds. When I look at a bird, I think that's a bird. And they found that when bird watchers are looking at birds and identifying different kinds of birds, this brain area is more active. Same thing is observed for people who are car experts, right? Some people know nothing about cars. When you look at a car, you think that's a car, and you don't really see the big differences between different kinds of cars. Other people are into cars, they can recognize different kinds of cars, and when their brains are scanned, we see activity in this area. In a famous study run shortly after the original sort of fusiform face area study came out, arguing that this brain area is face-specific, Guthrie and colleagues had participants distinguish between greebles. So these are made-up beings that have a small number of common parts. And they looked at the response in the fusiform face area, and initially the response in these individuals was quite high for faces, as you would expect, and it was quite low for greebles. Then they gave these individuals extensive training. They basically trained them on how to distinguish one greeble from another. So how to distinguish this greeble from this greeble based on its facial features. And what they found was that after training, this brain area responded much more strongly to greebles. It actually responded as strongly as it did to faces. So this is some evidence that this particular brain area, it's not that it's a face-specific brain area, but it seems to reflect acquired visual expertise. So it seems to reflect our ability to have expertise in visual recognition. Okay, we've come to the lecture review. Question 1. Neurons in V1 have small receptive fields that respond to contrast changes in edge orientation. How do neural responses to visual stimuli change as we move from V1 into the temporal lobe? So, how do neural responses to visual stimuli change as we move from V1 into the temporal lobe? The neurons respond to more complex images, and they have larger receptive fields, so they're excited by bigger patches of retina. Question 2. DF suffered damage to her temporal lobe. What was unusual about patient DF's behavior? So think about that mail slot study. What was unusual about patient DF's behavior? What was the deficit that Mel Goodall observed? So question 2, what was unusual about the patient DF's behavior? She could interact with objects that she failed to recognize, that she failed to perceive. And this was some evidence, right, that the brain distinguishes between vision for recognition and vision for action. What are the functions of the dorsal and ventral streams? I may have just given it away. Question 3, what are the functions of the dorsal and ventral streams? The dorsal stream allows us to interact with objects, while the ventral stream allows us to recognize and identify objects. So vision for recognition versus vision for action.